Lightning Biologists. In this video, I will go through the Practicing DNA Transcription and Translation worksheet that is posted. If you have the ability to print that off, that's fantastic. If you don't, you can simply write the sequences on a piece of paper using the worksheet as a template on your computer. So starting, let's just really quick review the two processes that we are going to look at. Number one is transcription. And if you think about transcription, transcription is taking the information on our DNA molecule and changing that into messenger RNA. Messenger RNA can then leave the nucleus, where the process of translation can happen out in the cytoplasm or in the rough endoplasmic reticulum at a ribosome. And that is where the messenger RNA will be further decoded into what we know as a protein. And that protein is a chain of amino acids. And so you can see here is a codon chart. And inside there are a list of all the amino acids that are possible to be strung together to make up all of the proteins that we know exist in all living organisms. So this is why we say DNA is the blueprint of life, because life basically is the makeup of the proteins, and therefore the code for those proteins is found in the DNA. So let's get started. We'll work on example 1, and I'll just walk you through example 1. We start with our DNA, and this is in the nucleus. And you can see that this worksheet is nice because it broke it into every 3 nitrogen bases. And the reason it does that is because we actually read messenger RNA 3 bases at a time, and we call that a codon. And so in looking at DNA, we have to think back to what are the base pair rules. We know that A will always bond with T, and we know that C will always bond with G. And those base pair rules you have to memorize. Now, we also know that this is for DNA. However, something changes when we go to RNA, and that is that we don't have thymine in RNA. In RNA, that thymine then gets replaced with U for uracil. And so when we're making RNA, we have to make sure that we don't ever place a thymine in a strand of RNA, because that would be incorrect. So let's just start. Thinking back to the base pair rules, if I have DNA, it's in the nucleus, the RNA polymerase has opened the double helix up, and now we're reading the strand of DNA, and the RNA polymerase is going to call in the complement nitrogen base that you would find in RNA. So it's the RNA nucleotides that RNA polymerase will call in. And so we're going to read, it's going to be T. So we know that T, its complement would be A. So the first nucleotide that's going to come in is going to be an A. RNA polymerase will see the A, and it will know to call in a U, because we're making RNA, and U complements A. The next one is a C, and so it'll call in a G. RNA polymerase will continue to move down the strand of DNA, and it will continue to bring in the complements. So we have a C, it'll bring in, there's a C on the DNA, so its complements a G, C. Here we have a C, so we'll bring a G in, a G. Here we have a T, so it'll complement to A. A, we're making RNA, so it has to be U, C, C. So you notice every nitrogen-based letter, I'm adding the complement that would bond to it. So if it's a G, guanine, it's always going to be cytosine. Cytosine, cytosine. The T always connects with A, adenine, cytosine, cytosine. So now I have my messenger RNA. I have actually completed the process. So from DNA to messenger RNA is the process of transcription. Transcription took place in the nucleus, and now the messenger RNA can take that information in the DNA out of the nucleus to the ribosome, where it can be decoded into an amino acid chain, which will become the protein. Here's where now we have to start looking at these 3 bases at a time. These 3 bases at a time are known as a codon. And we have to decode that codon into a chain of amino acids. Here's where we use our codon chart. So you always use messenger RNA. Later on, I'm going to ask you to tell me what the anticodon on the transfer RNA would be, but you don't ever use that when we come back to the codon chart. So if we look at the codon chart, and we'll come back up here, I'll change the color just to make it a little bit more user-friendly. The amino acids. So now I'm going to read codon at a time. So the first codon is AUG. If I look up here at the codon chart, I see that there is a first position, a second position, and a third position. That's talking about the bases in the codon. So the first position, which is right here on this side, so these are the first position. We're going to come down to the codon, and we see that there's an A. So first position, there's U, C, A, G. Those are all the possibilities. But in our codon, it's A. And so I'm going to be able to reduce my codon chart down to this row here, because these are all the possibilities if A is in the first position. And in our codon, it's AUG. In the second position, we go to the top. In the second position, we now have to come down to our codon. In our second position, it's the U. And so I find right here's my U. And so I'm going to go down, and where U crosses A, so right here in this box, I can now isolate the amino acid that this codon's coding for to this box right here. I do need the last or the third position nitrogen base in my codon in order to figure out, is it isoleucine or is it methionine? And so in this case, my third position nitrogen base, third position's over here, is a G. And so G's right here. So G I'm going to bring all the way across, and unfortunately it's kind of the same color. So I'll go ahead and I will circle methionine. It's the A, the U, and the G that brings us to that single amino acid, methionine. Now, I don't want to write out the whole word methionine, and so I am simply going to write the first 3 letters, MET. That will be the first amino acid that this messenger RNA strand is coding for. All right, now we're going to go to the second codon. We're going to do the same thing. So I'll come up here and I'll erase what I have so we can start over. And in this case, my first position is C. So I'm going to come and find C, and I'm going to narrow it down to this row. My second codon, or my second nitrogen base in my codon, is a G. And so I'll come over here, I'll bring this down, and you'll see that I now have it narrowed down to this box. And this is interesting because it doesn't matter what my third position is. My second position was a G, and so anything that starts CG actually has arginine. You would never have to memorize this codon chart. I worked in DNA, sequencing DNA for over 4 years, and we always had a codon chart available. So please do not try to memorize a codon chart. In this case, then, we will go to the third position just to help us understand it. The third position's over here. It's a C, and so in that situation, if I bring this over, you will see that it is arginine. And I'll come back down here, and I will write the first 3 letters, A-R-G. I'll continue to move over. At this time, what I'd like you to do is stop the video and continue and complete example 1, and then come back and start the video to check your answer. Okay, your first chain of amino acids should read methionine, arginine, glycine, serine, proline, and threonine. Let's move on to example number 2. In example number 2, we will start with DNA in the nucleus again. And go ahead and let's transcribe DNA into messenger RNA. Again, base pair ruling. If it's TTC, then the complement will be AAG. Go ahead and continue on. All right, once you have the messenger RNA transcribed, now the messenger RNA is going to leave the nucleus and it's going to go out to the ribosome. In this example, we've added an additional step, and that is for you to tell us what would be the RNA sequence of the transfer RNA that is going to bring the amino acid in. And so if you think back, the messenger RNA is going to go to the ribosome. At the ribosome, you're going to have this molecule that kind of looks like a T. At the top of that molecule, it's actually going to be carrying an amino acid. So here's your amino acid. And at the bottom of the molecule is where you are going to find your anticodon. And so there will be 3 bases, for example, AUG. This is called the anticodon. The anticodon is anti the codon, but not really, because it's actually the complement of the codon. And this is how the transfer RNA knows it has the right amino acid for the sequence of the messenger RNA. So if we go back to our messenger RNA and we look at the first codon, because codon is on messenger RNA, the transfer RNA would be carrying the anticodon. And in this case, the anticodon is going to be the complement of AAG, which would be UUC. We don't use the transfer RNA anticodon in order to determine the amino acid. We actually will always use the codon to determine the amino acid because that's how we create the codon chart. It's easier, it skips a step. But let's go ahead and shut the video off and see if you can fill in the transfer RNA that would come in and bring the amino acids for this particular example 2 strand of messenger RNA. There you have the transfer RNA that you can check your answers against. And now we'll go back to using the messenger RNA codon to actually determine the amino acid. And again, just as a quick review, we're going to use AAG. So we're going to go back up to our codon chart. We're going to find the A. So we know it's in this row. The second one was also an A, and so we know that it's here. And the last one, if I go back and check, it was a G, and so the G is right here. And so you are going to see it is lysine. Once you have example 2 and 3 done, you can check it here with the answers. Good luck.